So picture a cricket, just your standard, ordinary forest cricket,
Yeah.
right?
Just a normal bug.
Exactly. It's living its life on the dark, damp forest floor. And it's a nocturnal creature, so it's perfectly camouflaged in all these mottled browns and greens.
Yeah, it wants to blend in.
Right. It spends its entire existence basically hiding in the low brush, just trying to avoid predators. And one thing it absolutely does not do is swim.
No, definitely not. Crickets and water, they do not mix at all.
Yeah, they lack the buoyancy and the physical structure for it. They avoid large bodies of water at all costs. But then on this warm summer night, the specific cricket emerges from the brush.
It just comes right out into the open?
Yeah, it starts moving erratically, leaving the safety of the shadows. And it actively seeks out a brightly lit open space, specifically a swimming pool that's just glowing turquoise in the moonlight.
Which is basically the most exposed, dangerous place it could possibly be.
Exactly. And then defying every single survival instinct programmed into its DNA by like millions of years of evolution, the cricket just takes a flying leap. It plunges headfirst into the water to drown.
It's just, it creates a deeply unsettling image, honestly.
It really does.
Because watching an animal commit what looks like deliberate, unprompted suicide, I mean, it forces you to question everything you know about biological imperatives.
Right, because the primary directive is survival.
Exactly, survival and reproduction. So when an organism actively destroys itself like that, the natural order just feels momentarily broken.
Well, you sent us this massive stack of research and articles and personal notes on this exact phenomenon. And as we were poring over your materials, we realized how fundamentally this flips our traditional understanding of illness.
Well, it completely turns it upside down.
Because normally when we think about a disease, we think of it like a car breaking down. You know, the engine sputters, a tire goes flat, the transmission drops, and the car just simply stops moving.
Right, and biologists call that pathology. The organism is just sick or exhausted. It's physically broken by an invader.
But looking at your notes, that is not what is happening to this cricket. This isn't the car breaking down. This is a scenario where someone has hacked the car's GPS, locked the doors from the inside, and is deliberately steering the vehicle off a cliff.
That is a perfect way to put it.
Yeah.
And that distinction between pathology and manipulation is the core of what we're unpacking today.
Because we like to think we're in the driver's seat, right?
Oh, absolutely. We like to think we are completely alone in the driver's seat of our own minds. That we are completely in control of where we steer, what we desire, what we fear.
But the reality of this research suggests something else.
Yeah, it suggests there might be an invisible passenger in a lot of biological systems. And that passenger might have their foot pressing down very, very hard on the accelerator.
So today, for the listener, we are taking a deep dive into the world of parasites. And we've custom-tailored this specifically for you. We're not just talking about the biological hitchhikers that give you a rash or a fever.
No, we are talking about master manipulators.
Right. Microscopic puppeteers that literally hijack complex nervous systems. And our mission for this deep dive is to explore exactly how they pull this off on a molecular level.
Yeah, how they engineer entire food webs.
Exactly. And perhaps most surprisingly, how they might subconsciously be driving human culture. You know, our societal norms, our political beliefs, and even the daily decisions you make.
And to really appreciate the sheer scale of this invisible influence, we have to recognize a massive paradigm shift that happened in science.
Because they didn't always look at parasites this way, right?
Not at all. For most of the last century, the scientific community completely ignored the idea of parasite intelligence.
Really? Completely ignored it.
Yeah, basically universally.
Mm.
Because these organisms are so small, and because they can't survive independently outside a host, biologists just view them as degenerate, primitive simpletons.
Like they were just lazy.
Exactly. They were considered evolutionary dead ends that had somehow lost the ability to hunt or forage for themselves.
They were viewed as the ultimate biological moochers, just looking for a free ride.
Yes, the academic consensus dismissed them as evolutionary failures. But what we realize today, thanks to the emergence of this completely new interdisciplinary field called neuroparasitology, is that this reliance on a host isn't a weakness at all.
It's an advantage.
It is an evolutionary superpower. They haven't lost their abilities. They've just outsourced the heavy lifting of survival to larger, more complex animals.
Which is brilliant when you think about it.
It really is.
Yeah.
I mean, why build a complex digestive system or a massive musculoskeletal structure when you can simply invade an organism that already has one
Yeah.
and then just control it from the inside out?
Wow. So to really grasp how these manipulations work, we first have to understand why it took science so long to even admit this was
Oh,
happening.
the history is fascinating.
It really is. Going through your notes, it's this story of academic stubbornness and rigid compartmentalization. And the journey really starts in the 1970s with a researcher named Janice Moore.
Yeah. Janice Moore serves as a foundational figure in overcoming that intense academic stigma. Let's look back at 1971. She was a senior at Rice University in Texas, sitting in a parasitology class.
Taught by a pretty famous professor, right?
Right, Clark Reed. He was highly commanding, incredibly eccentric. He would stand at the front of the lecture hall just puffing on cigarettes, telling these rich, almost mysterious stories about the hidden lives of parasites.
I love that image.
And during one lecture, he threw out a challenge to the class. He just said, think like a trematode.
Okay, for context, a trematode is a type of parasitic flatworm, right?
Exactly. So Reed described this one specific trematode that spends part of its early life cycle inside an ant. But it has a massive logistical problem. It can only sexually reproduce inside the bile duct of a grazing sheep.
Wait, let me stop you there. A sheep grazing on grass is basically a giant woolly vegan lawnmower.
Right.
It has absolutely zero interest in hunting or eating insects. So if the parasite is stuck inside an ant, it's effectively stranded, right?
Logically, yes. The logistical nightmare for the parasite is figuring out how to cross this massive biological void between an ant colony and a sheep's digestive tract.
So how does it do it?
Well, Reed explained to his class that the trematode solves this by physically invading the ant's brain.
Oh, wow.
Where once night falls, instead of returning to the subterranean colony with the rest of the worker ants, the infected ant just breaks rank. It climbs to the very top of a blade of grass and clamps its jaws shut onto the tip.
Just locks on.
Exactly. It enters a state of lockjaw. And it just dangles there in the darkness, waiting to be accidentally consumed by a sheep grazing in the cool night air.
It is like a comic book supervillain overriding a robot with a joystick.
It really is.
But the part of the story in your notes that blew my mind is the timing mechanism. Because if the sheep doesn't happen to eat that specific blade of grass by morning, the ant is in severe danger.
The timing is the truly brilliant evolutionary adaptation here. Because when the sun comes up, the ambient temperature rises. If the ant stays at the top of the grass blade during the midday heat, it will literally bake to death in the sun. And if the host ant dies from heat exposure, well, the parasitic trematode trapped inside it dies too.
So what does the parasite do?
It senses the temperature shift and literally releases its chemical grip on the ant's brain.
You're kidding.
No, it lets the ant unclench its jaws, climb back down to the ground, and act like a totally normal ant during the dangerous heat of the day.
That is wild.
And then once the sun sets and the air cools down again, the parasite seizes control, again, forces the ant back up the grass blade, and clamps the jaws shut once more. It only exposes the ant during the cool, safe grazing hours.
That is terrifyingly precise. I mean, it's reading the environmental temperature and turning the host's free will on and off like a light switch.
Exactly.
So Moore hears this in class, and she's completely hooked. She decides she wants to dedicate her life to studying this phenomenon. But she immediately ran into a brick wall of academic hostility.
She faced a massive wall of resistance. When she moved on to graduate school at Johns Hopkins, she discovered that the scientific world was incredibly stubbornly compartmentalized.
Like different departments just didn't talk.
Right. The ecologists studying environments didn't talk to the parasitologists studying worms. The neuroscientists studying brains didn't talk to the behavioral biologists.
It was all siloed.
Completely.
Yeah.
There's this story where during one seminar, a really respected marine biologist was giving a highly complex, mathematically dense lecture on the movement patterns of aquatic snails.
Okay.
And Moore just stood up in the audience and simply asked if he had checked his subjects for trematode parasites. She pointed out that infected snails behave radically differently than healthy ones.
And according to the historical accounts, the guy got genuinely angry with her.
He was visibly furious. He had spent years tracking water currents and ambient temperatures and predator populations to build his model.
And she's basically saying his data might be corrupted by worms.
Right? The suggestion that a microscopic invisible worm was secretly pulling the strings, rendering his complex environmental models useless, that was an incredibly unwanted complication.
I can imagine.
Moore actually ended up dropping out of Hopkins because the resistance to this idea was so deeply entrenched.
Because the default assumption was just sickness, right?
Exactly. The default was always pathology. If an animal was acting weird, it was just sick. For instance, years later, one of Moore's own graduate students did a study showing that dung beetles infected with roundworms dug significantly shallower burrows.
Which would alter the soil aeration of the whole local ecosystem?
Yes, perfectly. But when they submitted the paper to a prestigious journal, the editor rejected it outright.
Just no peer review at all.
No peer review. They wrote back that the shallow digging was obviously just a, quote, side effect of illness and lethargy, not a deliberate manipulation by the worm.
Wow, that academic blindness is just staggering. Which brings us to another incredible story of scientific rebellion from the sources you provided. We have to talk about the French evolutionary biologist, Frédéric Thomas.
Oh, yes.
He was the researcher tracking those suicidal crickets we talked about at the very top of the deep dive.
The story of Frédéric Thomas perfectly illustrates what it takes to break a scientific paradigm. In the late 1990s, Thomas wanted to travel to New Zealand to study these crickets that were inexplicably throwing themselves into water.
Okay, so he applies for a grant.
Right. He applied for a standard research grant from the CNRS, which is the French National Research Agency. And keep in mind, Thomas was not a fringe scientist. He was a recognized prodigy. He had 14 peer-reviewed publications to his name already.
But the agency rejected it.
Flatly rejected his proposal. The reviewers stated they simply didn't see the scientific or financial value in studying an insect acting against his own survival interests.
They just thought it was a fluke.
Yeah, they viewed it as a freak anomaly, not a systemic biological process.
And his reaction to that rejection was definitely not standard academic protocol.
Not at all. He didn't just write a polite appeal or complain to his colleagues. He drafted a letter directly to Jacques Chirac, who was the president of France at the time.
That is a bold move.
And in the letter, Thomas threatened to go on a highly public hunger strike if his research grant wasn't funded. He argued that the French government was stifling groundbreaking evolutionary biology.
You have to respect the sheer audacity of that commitment, and the crazy thing is it actually panicked the government bureaucracy.
It did. They sent regional officials down to his university in Montpellier to pressure his department head. They basically threatened to pull funding from the entire biology department if they couldn't get this rogue young scientist under control.
Incredible. So did he strike.
Well, Thomas eventually backed down from the hunger strike because he managed to secure private funding from a sympathetic Swiss billionaire. So he takes the money and flies all the way to New Zealand.
But ironically, nature didn't cooperate.
No, they couldn't find enough infected crickets in the New Zealand forests to conduct a proper study.
So Thomas comes back to France, frustrated and empty-handed. But the twist of fate in your notes here is just remarkable.
It's almost unbelievable. Shortly after he gets back, he's talking to a lab technician who casually mentions that his cousin cleans swimming pools at a vacation resort.
Just 75 kilometers from Thomas's home.
Right. And the cousin was constantly complaining about pulling these massive, disgusting worms out of the pool filters, and they were always tangled up with drowned crickets.
So Thomas takes his wife to this romantic resort, supposedly for a weekend getaway. But instead of enjoying a romantic evening, he sneaks out to the glowing swimming pool at night, armed with test tubes and a flashlight.
As one does.
Right. And he literally watches a cricket march out of the bushes, step up to the edge of the pool, jump in, and as the insect drowns, this massive 3-inch-long nematode worm comes squirming out of its body.
The exact phenomenon he flew across the globe to find was happening right in his own backyard in France.
Which forces us to ask a really crucial foundational question. If this behavioral manipulation is happening right in front of us, in resort swimming pools, on blades of grass, in sheep pastures, in the soil beneath our feet, why were scientists so stubbornly blind to it for over a century?
Yeah, why couldn't highly trained observers see what was literally right there?
Exactly.
Well, Janice Moore provides a brilliant philosophical answer to this. We are imprisoned by our senses.
What does she mean by that?
We only look for what we are biologically capable of perceiving. She uses the historical analogy of bat echolocation to explain this blindness.
Oh, I remember reading this part.
Yeah, for 150 years, scientists knew that blindfolded bats could fly perfectly through a room full of obstacles. But if you plugged their ears with wax, they crashed.
So they obviously knew hearing was involved.
You would think so. Yet the scientific establishment absolutely refused to believe that bats were navigating by sound, simply because human ears couldn't hear the high-frequency ultrasonic clicks the bats were making. Because the scientists couldn't hear it, they assumed it wasn't happening. They attributed the bat's navigation to some mystical sixth sense or air pressure sensitivity.
So until we had the tech to hear it, we just denied it.
Exactly. It wasn't until military sonar and radar technology were declassified after World War II that biologists finally had the mechanical instruments to hear the bats and accept echolocation as a fact.
Because we couldn't experience it with our human hardware, we arrogantly assumed it didn't exist in nature.
Precisely. And Moore and other neuroparasitologists point out that we currently only notice the parasitic manipulations that happen to be visually obvious to human eyes.
Like a cricket jumping into a pool.
Right, or an ant dangling on a grass blade, or a mouse running toward a cat. But what if a parasite alters a host's behavior via invisible pheromone scent trails?
We would never know.
Exactly. What if it subtly changes the host's internal body temperature to make it seek out different microclimates? What if it makes the host emit acoustic signals far beyond human hearing? Or exposes body parts that reflect ultraviolet light patterns that only predatory birds can see?
So there could be a whole world of mind control happening that we literally cannot perceive.
We are likely missing tens of thousands of complex manipulations every single day simply because our human sensory toolkit is entirely unequipped to perceive them.
That completely shifts how you look at the natural world. I mean, every insect, every bird, every fish could be operating under an invisible set of instructions.
Yeah, it's a humbling thought.
But now that the scientific community has finally admitted this is a real and pervasive phenomenon, we need to look at the actual mechanics.
Yes, the how.
Right. How exactly does a microscopic single-celled blob or a tiny worm physically override a complex multi-layered nervous system?
It's a step-by-step process.
So let's frame this section like a masterclass for any biology students listening, a step-by-step educational tutorial on how you identify and break down parasite-induced behavioral changes.
I love that framework. So step one of the tutorial, identifying the evolutionary motive.
The why.
Exactly. A parasite does not expend massive amounts of metabolic energy to control a host just for fun or malice. The manipulation always serves one singular goal: transmission.
Moving from host A to host B.
Right. It has to complete its life cycle and reproduce. If we look at the trematode we discussed earlier, its ultimate motive is to get out of the ant and into the sheep's bile duct. Everything it does neurologically to the ant is reverse engineered from that required endpoint.
Okay, so once the motive is established, the parasite has to physically move the host. Step 2, altering locomotion and timing. With the ant, we talked about how it physically forces the clamping of the mandibles onto the grass.
Yes.
But how is it doing that? Is it severing nerves? Is it destroying muscle tissue?
It is much more elegant than destruction. If a parasite destroys the host's motor functions, the host becomes paralyzed and useless.
Right, it wouldn't be able to climb the grass.
Exactly. Instead, the parasite influences the neuromuscular junctions. In the case of the ant, the parasite migrates to the subesophageal ganglion.
So what?
It's a cluster of nerves that controls the ant's mouth parts. It doesn't destroy those nerves. It applies physical or chemical pressure that triggers a localized tetanus-like state, forcing the muscles to contract and lock.
Oh, wow.
And as we mentioned, it pairs this physical lock with temperature sensitivity, ensuring the ant isn't killed by the midday sun. It's an incredibly delicate balance between hijacking the vehicle and keeping the engine running.
That brings us to step 3, which is where it gets highly molecular. Hacking perception and neurochemistry. Let's go back to Thomas's suicidal crickets and the hairworm.
Right, the pool jumpers.
Yeah, how does a worm inside the abdomen make a cricket's brain want to drown itself? I saw in your notes a molecular biologist named David Biron tackled this.
He did, using a technique called proteomics.
Let's break down proteomics for the listener because it's a vital concept. Think of the cricket's brain like a massive, highly secure chemical factory. Proteomics is basically a method where scientists sneak in and read the factory's daily shipping manifest, right?
That is an excellent analogy, yes.
They want to see exactly which proteins and chemicals are being manufactured and in what quantities at any given moment.
So Byron analyzed the protein manifest of infected crickets versus uninfected crickets.
Yeah.
And what he found was astonishing. He discovered that the hairworm is literally manufacturing complex neurochemicals that perfectly mimic the cricket's own biological language.
It's speaking cricket.
Exactly. The worm is producing proteins that the cricket's central nervous system recognizes as its own internal commands. It creates what scientists call molecular crosstalk.
So the worm isn't just passively damaging the brain, it is actively talking to it, issuing fake chemical orders that override natural instincts.
Yeah.
And what specific orders is it giving? How does it make the cricket jump into the pool?
Well, Biron's proteomics analysis revealed that the worm dramatically increases the production of proteins specifically related to the cricket's visual processing processing centers.
It messes with its eyes.
It fundamentally alters the insect's optical perception. A normal cricket is photophobic. Its brain tells it that light equals exposure and death, so it seeks the dark.
Right.
The worm's chemical cocktail reverses that visual polarity. It makes the cricket intensely, irresistibly attracted to the brightest thing in its nighttime environment.
Which in a natural forest setting is moonlight reflecting off the surface of a stream or a pond. Or in modern France, a glowing turquoise swimming pool.
Exactly. The worm is literally hacking the cricket's optical hardware to turn a fear of light into an addiction to it.
Okay, so that's internal chemistry. But step 4 in our tutorial is the physical makeover. Sometimes a parasite doesn't just change the chemistry, it executes a massive physical alteration so the host attracts the next predator.
Yes, and the classic, perhaps most visually disturbing example of this is the Leucochloridium flatworm, which infects amber snail.
This example from your research is purely wild. It sounds like something out of a science fiction horror movie.
It truly does. The flatworm needs to get from the snail into the digestive tract of a songbird to reproduce. To do this, the worm's larvae invade the snail's tentacles, its eye stalks.
Oh boy.
As the larvae grow, they pack themselves into specialized brood sacs. These sacs stretch the skin of the snail's eye stalk so incredibly thin that you can actually see the parasite moving inside.
That is so gross.
But the parasite doesn't just sit there. It forms these brightly colored green, white, and red bands that begin to pulsate, rhythmically beating at up to 70 times a minute.
It is basically installing a neon strobe light on the snail's face.
Exactly. But a strobe light hidden deep in a dark bush doesn't help. So simultaneously, the parasite alters the snail's behavior. Snails normally hide deep in the undergrowth to avoid the sun and predators.
Let me guess it makes them seek light.
The flatworm chemically suppresses the snail's aversion to light, forcing it to crawl out of the dark shadows and up onto the highest exposed leaves in broad daylight.
So to a hungry songbird flying overhead, those pulsating, colorful, swollen eye stalks sitting on a high leaf look exactly like fat, juicy, wriggling caterpillars.
And the bird swoops down, bites off the eye stalks, and the parasite successfully enters its final host.
And the most gruesome part, the poor snail often survives the attack. It falls back into the bushes, slowly regrows its eye stalks over several months, and because it lives in an environment contaminated with bird droppings, it likely gets infected all over again.
It is trapped in a cyclical nightmare.
Wow. Okay, so getting into the next host is only half the battle. Let's look at step 5, the ultimate escape artist. What happens if the timing of the heist goes wrong? Let's go back to Thomas's suicidal cricket. When the cricket jumps in the water, it isn't always finding an empty swimming pool.
No, usually it's a forest stream.
Right, and a Saracen cricket usually gets eaten by a fish or a frog immediately. If the worm's goal is to lay eggs in the water, does it just die inside the frog's stomach? Is the whole manipulation a failure?
This specific question plagued Frederick Thomas. So he conducted an experiment. He went to a local pet store, spent 25 euros on a predatory frog, put it in a glass aquarium in his lab, and tossed in a cricket infected with a hairworm.
Okay.
As expected, the frog lunged and snapped up the cricket, instantly swallowing it whole. Thomas sat back incredibly confused, because if the hairworm is digested along with the cricket, the entire elaborate evolutionary strategy of driving the cricket to water is a dead end.
But the worm doesn't get digested, does it?
Oh, it doesn't. Thomas watched in absolute shock as a few minutes later, the massive 3-inch hairworm casually squirmed back up out of the frog's mouth, dropped into the water, and swam away.
No way.
It turns out after being swallowed, the worm realizes it is in a highly acidic, hostile environment. It immediately escapes the dying cricket's body, inside the frog's stomach, turns around, and travels all the way back up the frog's esophagus against the muscular contractions of swallowing.
It just climbs back up.
Sometimes it even bypasses the mouth and squeezes out through the frog's nostrils. And if the cricket happens to be eaten by a trout or a predatory fish, the worm quickly navigates up the digestive tract and escapes out through the fish's gills.
Let's just pause and appreciate the sheer evolutionary audacity of that. Imagine you are walking through the jungle, you get swallowed completely whole by a lion, and instead of being dissolved by stomach acid, you just casually stroll back up the lion's throat, walk out of its nose, and go about your day.
It is an unmatched level of resilience.
It is breathtaking. And if we connect this microscopic resilience to the macroscopic world, it forces us to completely rethink how entire ecosystems operate. Which brings us to the next massive paradigm shift in your research.
Ecosystem engineers.
Right. If parasites are physically steering prey into the mouths of predators, serving them up on a silver platter, then our fundamental understanding of ecosystem dynamics and food webs is deeply flawed.
It absolutely is.
We've all seen those high-budget nature documentaries. You know, the soaring orchestral music plays, the camera goes into ultra-slow motion, and you watch the, quote, supreme apex predator, a hawk, a shark, a lion swoop down with majestic grace to catch its prey.
It looks like a pure triumph of strength and agility.
But the reality exposed by neuroparasitology is that the majestic predator might just be eating the slow, the dumb, or the neurologically hacked. They aren't executing a brilliant hunt, they are eating low-hanging fruit that was specifically delivered to them by a parasite.
Let's look at the specific empirical data that proves this. An ecologist named Kevin Lafferty spent years studying a type of fluke parasite in the complex estuarine ecosystems of Southern California.
Okay, so what's the life cycle of this fluke?
Well, it lives in the guts of shorebirds, passes its eggs into the mud via bird droppings, infects tiny horned snails grazing in the mud, and then swims out of the snails to infect California killifish.
Then what does it do to the fish?
The parasite larvae physically migrate to the killifish's brain, caking themselves onto the surface in a dense layer.
But initially, Lafferty didn't see anything physically wrong with the fish, right? They didn't have strobe light eye stalks or obvious deformities?
Right to the naked eye, the fish looked totally healthy, swimming alongside uninfected fish. But Lafferty had an undergraduate assistant, Kimo Morris, who spent hundreds of hours simply observing the fish in laboratory tanks.
And he noticed something.
Morris eventually noticed a subtle behavioral anomaly. He realized that the infected fish were darting to the surface of the water and suddenly rolling over on their sides. In fact, they were performing this rolling maneuver 4 times more often than the healthy fish.
Now, context is everything here for the listener. Killifish have bright silver, highly reflective bellies, so by darting to the surface and rolling over, they are essentially taking a mirror and flashing sunlight directly up at the sky.
Exactly. They are acting as visual beacons. They are actively signaling the predatory shorebirds circling overhead. The birds see the flash dive down and eat the fish, completing the parasite's life cycle. Lafferty's research demonstrated that the parasite is acting like a giant invisible conveyor belt. It is actively moving biomass energy from the mud of the estuary up into the fish population and ultimately feeding it into the sky to sustain the bird population.
That is brilliant. And there's another incredibly vivid example from your notes. It involves a tapeworm that infects tiny translucent brine shrimp.
Oh, this one is visually spectacular.
The tapeworm completely reconstructs the shrimp's identity, right?
It does. The tapeworm chemically castrates the brine shrimp, effectively stealing the energy that shrimp would normally use for reproduction to fuel its own growth. It alters the shrimp's lifespan, making it live longer to ensure the parasite matures.
Okay.
But most importantly, it changes the shrimp's color from transparent to a bright, vibrant, opaque pink.
It paints them pink.
Basically, yes. It then chemically overrides the shrimp's natural instinct to stay solitary and hidden, forcing them to swarm together in massive, densely packed groups near the water's surface. You can fly over these lakes and literally see massive red clouds in the water that are over 1 meter across.
They're forming a giant glowing neon sign in the water that says, "Eat me."
And who is the target audience for that sign?
Flamingos.
Flamingos. They are filter feeders. They don't want to spend all day chasing individual transparent shrimp. Thanks to the parasite, the flamingos don't have to hunt at all. They just wade over and dip their beaks into this dense, brightly colored, pre-packaged seafood broth that the tapeworm has conveniently gathered for them.
That is just wild to me. So you're saying this tiny bug is calling the shots for an entire flock of flamingos?
The parasite must enter the flamingo to complete its life cycle, so it makes the shrimp an irresistible, effortless meal.
There is an amazing real-world consequence to this dynamic, too. Your notes highlight a team of Japanese biologists who were desperately trying to save a highly endangered species of river trout.
Yes, this is a great example of ecosystem engineering.
For years, they couldn't figure out how these specific trout were surviving the late autumn season. Because during autumn in these Japanese forests, the aquatic insects the trout normally eat die off, and food becomes incredibly scarce.
By all ecological models, the trout population should have starved to death.
So what did they find?
The biologists decided to analyze the stomach contents of the trout during the starvation period, and to their absolute shock, they found the stomachs packed full of terrestrial crickets and grasshoppers.
Which makes zero ecological sense because crickets live in the forest and avoid water at all costs.
Right. But upon further investigation, it turned out that a hairworm, closely related to the one Thomas studied in France, was heavily infecting the local insect population. The worms were driving massive droves of crickets to drown themselves in the streams exactly at the time of year when the trout were facing starvation.
So an invisible parasite literally engineered the survival of a massive endangered apex predator.
Yes.
The worm was the primary caloric input keeping the trout from extinction. The birds, the flamingos, the trout, none of them are independent operators.
That is the crux of neuroparasitology's impact on ecology. Parasites are the ultimate ecosystem engineers. They don't just exist passively within a habitat, they actively restructure the flow of energy and biomass.
Okay, let me play devil's advocate here and push back on this. We spend billions of dollars globally on human medicine, veterinary science, and agriculture trying to eradicate parasites. We view them as a scourge.
Sure we do.
If we somehow possessed a magic wand tomorrow and we could instantly cure every animal on Earth of all parasitic infections, wouldn't the ecosystems be objectively healthier? Wouldn't the animals thrive without these things eating them from the inside out?
It is a logical assumption, but the ecological reality is the exact opposite.
Really?
If you waved that magic wand, you would likely cause catastrophic ecosystem collapse globally. Because parasites are acting as that conveyor belt, removing them would completely sever vital links in the food chain.
Oh, because the predators are used to the easy meals.
Exactly.
Yeah.
Predators have evolved over millions of years to rely heavily on that easy parasite-delivered biomass. The birds rely on the flashing killifish. The flamingos rely on the swarming shrimp. The trout rely on the suicidal crickets.
Just without them.
Without the parasites serving up the prey, the predators would have to expend significantly more energy to hunt healthy evasive prey. Many predator populations simply wouldn't be able to adapt fast enough, and populations of birds, fish, and mammals would crash. The entire architecture of the ecosystem is load-bearing on the backs of parasites.
That completely upends how you view nature. But it's not like the host animals are just passively sitting there resigning themselves to being manipulated and eaten. There's a massive silent war going on, isn't there?
Absolutely.
A multi-billion-year evolutionary arms race between parasitic cunning and host defense.
The hosts are constantly evolving countermeasures, and organisms fight back against parasites in two primary ways: physically and psychologically.
Let's start with physically. We see constant active behavioral defenses in nature that we often mistake for just quirky animal habits, right?
Yes. When dogs lick their wounds, they aren't just cleaning dirt. Canine saliva contains specific bacteria-killing compounds. Blue herons, standing still in marshes, are plagued by mosquitoes. Biologists have recorded them pecking at the mosquitoes, hovering beneath their feathers up to 3,000 times an hour.
3,000 times an hour.
An incredible expenditure of energy purely for parasite defense. In Nepal, researchers observed wild elephants breaking off branches with their trunks, carefully stripping the side leaves to create a smooth switch, and using it as a specialized tool to swat away biting flies that carry blood-borne parasites.
They're literally manufacturing their own fly swatters to avoid infection. But the deeper systemic evolutionary defense is what evolutionary biologists call the Red Queen hypothesis, right?
Right. The Red Queen hypothesis takes its name from the character in Lewis Carroll's Through the Looking Glass. In the book, the Red Queen tells Alice, it takes all the running you can do to keep in the same place.
And how does that apply to biology?
It perfectly describes the evolutionary arms race. Parasites mutate and evolve incredibly quickly because they have massive population sizes and very short lifespans. They're constantly generating new genetic mutations, essentially figuring out new keys to unlock the cellular doors of their hosts.
So if the parasite is constantly forging new keys, the host organism has to constantly change the locks on its doors.
Exactly. And evolutionary biologists widely believe this is one of the primary reasons sexual reproduction evolved in the first place.
Really? Sex is about fighting parasites.
Well, asexual reproduction, cloning yourself, is incredibly efficient.
Yeah.
But it means your offspring have the exact same cellular locks that you do. If a parasite figures out how to invade you, it can instantly wipe out your entire cloned lineage.
Because they all have the same vulnerability.
Right. By mixing the DNA of two different parents through sexual reproduction, you shuffle the genetic deck for your offspring. It ensures the next generation has a slightly different set of molecular locks, forcing the parasites to start their complex lock-picking process all over again. We endure the massive biological costs of finding a mate and reproducing sexually, specifically to outrun parasites.
That is fascinating. But there is an even more immediate day-to-day defense mechanism that your notes highlight. It isn't happening at the cellular level, it's happening in our minds. The psychological shield known as the behavioral immune system.
The behavioral immune system is a relatively new and incredibly powerful concept in evolutionary psychology. The core idea is that long before your white blood cells ever encounter a physical germ, your brain is actively working to keep you away from it.
And the primary emotional driver of this system is the feeling of disgust.
Right.
Think about it. We don't have to be formally taught to be repulsed by certain things. If you smell rotting meat, or step in feces, or see a puddle of murky, stagnant green water, or sit next to someone who is visibly coughing and covered in skin lesions, your physical reaction is immediate.
You feel a visceral, gag-inducing wave of nausea. That feeling isn't arbitrary. It is an ancient evolutionary alarm bell. Your brain recognizes visual or olfactory cues associated with high pathogen loads and aggressively steers your behavior away from contagion.
It is a highly efficient, proactive defense system.
Yes, feeling disgusted by the smell of a dung heap keeps you 50 feet away from it, which protects you from thousands of different bacteria and parasites all at once without your internal immune system ever having to expend energy fighting them off.
So we've talked about crickets, snails, fish, and the broad concept of disgust. It is very easy to look at those animals and feel somewhat insulated. We are humans. We have massive cerebral cortices. We build cities. We have complex societies and language. We like to think we are above being hacked by a worm.
We really do.
But this is where the deep dive gets intensely personal. Let's turn our attention to the cat parasite, Toxoplasma gondii.
Toxoplasma gondii, commonly referred to as T. gondii, shatters the illusion of human invulnerability. It is a single-celled protozoan. Now, to be clear, a protozoan is a eukaryote, meaning it has a complex cell structure with a nucleus much more akin to a microscopic animal than a simple bacteria or a virus.
And its life cycle is tied to cats.
Its life cycle revolves entirely around felines. It can infect almost any warm-blooded animal, but it can only sexually reproduce inside the specific chemical environment of a cat's intestinal tract.
Okay.
The cat then sheds millions of the parasite's microscopic eggs called oocysts into the environment via its feces, where they can survive in the soil for months or even years.
And naturally, small rodents like rats and mice, while scavenging through the soil for food, come into contact with those feces and ingest the eggs, becoming intermediate hosts.
Now, consider the neurology of a normal uninfected rat. A rat is biologically terrified of cats. The faint scent of cat urine is deeply genetically hardwired into the rat's brain as a chemical signal that triggers the amygdala, the brain's fear center, telling the rat to freeze in place or run for its life.
Right, it's a pure survival response.
But researchers, most notably Joanne Webster in the UK and Jorislav Fleger in the Czech Republic, discovered something astonishing. When T. gondii enters the rat, it migrates directly to the brain and physically rewires those neural circuits.
When you say rewires, how exactly is it doing that? Like, does it just destroy the fear center so the rat becomes numb?
No destruction would make the rat lethargic and easy prey for non-feline predators like owls or snakes, which doesn't help the parasite.
True.
It is much more insidious. It doesn't delete the fear center, it bypasses it. The parasite alters the way the rat processes olfactory information. It takes the specific scent of cat urine and reroutes those neural pathways away from the amygdala and directly into the rat's sexual arousal and reward circuits.
It turns innate terror into physical attraction.
Exactly. The infected rat actively seeks out areas heavily marked with cat urine. It becomes hyperactive, loses its caution, and essentially parades itself around in the open, highly stimulated, until it inevitably gets noticed and eaten by the cat, thereby returning the parasite to its primary host to reproduce.
Fatal feline attraction. The parasite hacks the rat's brain to serve it up as a meal. But here's where this stops being a quirky story about rodents and becomes a global human health issue. Because cats don't just live in the wild.
No, they live with us.
They live in our houses, they sleep in our beds. How many humans are currently walking around infected with this exact same parasite?
The numbers are staggering. Depending on the country, local hygiene practices and dietary habits, anywhere from 10% to over 50% of the entire global human population currently has T. gondii cysts lying dormant in their brain tissue right now.
50% in some places.
In places like France or Germany, where eating raw or heavily undercooked meat-like seigneur beef or steak tartare is culturally popular, the infection rate frequently exceeds 50%. We're talking about billions of human beings carrying a brain parasite.
Billions of people have a cat parasite living inside their actual neurological tissue. For decades, how did the medical community view this?
For a very long time, the medical establishment viewed it as a completely benign, dormant infection, provided the human had a healthy immune system.
They didn't think it did anything.
Right. They acknowledged it was dangerous for pregnant women, which is why pregnant women are told not to change cat litter to avoid congenital transmission to the fetus, and dangerous for immunocompromised individuals. But for the average healthy adult, doctors assumed the parasite just sat quietly in a cyst doing absolutely nothing.
But that's changing.
However, modern neuroscience and advanced imaging are showing us a radically different, highly disturbing picture.
Let's look at the mechanics of how it operates inside a human brain. Your notes highlight the incredible work of a researcher named Patrick House. He discovered a mechanism that he nicknamed spit and run. What exactly is happening there?
House's research fundamentally changed how we view parasitic invasion. Traditionally, scientists thought a parasite had to fully enter and live inside a host cell to manipulate it.
Okay.
But House, using advanced microscopes and fluorescent genetic tags, watched T. gondii interact with living brain tissue. He discovered that the parasite can approach a human neuron attached to the cell membrane and inject a complex cocktail of chemical proteins directly into the cell and then detach and move on without ever fully invading it.
It's performing biological drive-by shootings on our brain cells.
Precisely. Under a microscope, House could see these altered injected neurons glowing neon green due to the fluorescent tags, and they were scattered widely across the brain tissue. The parasite doesn't have to establish a permanent base inside a cell to fundamentally alter its chemical behavior.
And what exactly are those injected chemicals doing? This leads us to the breakthrough from parasitologist Glenn McConkey, which honestly reads like a horror novel.
McConkey was analyzing the actual genetic code, the DNA of T. gondii, and he found something that shocked the parasitology world. The parasite possesses its own specific gene for manufacturing an enzyme called tyrosine hydroxylase, which is the limiting step in producing dopamine.
Let's pause and emphasize that for the listener. Dopamine isn't just a generic chemical. It is the core human neurotransmitter responsible for processing pleasure, reward-seeking behavior, fear responses, and attention. It is the chemical that makes us feel motivated or impulsive.
Yes, and a microscopic single-cell protozoan has the evolutionary genetic code to independently manufacture a complex mammalian neurotransmitter.
That is insane.
Once these parasitic cysts are established inside the brain, they act like tiny, unregulated dopamine factories. McConkey's team found that infected neurons were manufacturing up to 3.5 times more dopamine than normal healthy cells. This excess chemical was literally pooling inside the surrounding brain tissue, subtly shifting the host's neurochemistry.
Furthermore, your notes indicate it doesn't just stop at dopamine. It also impacts GABA and glutamate levels, right?
That's a critical point. To oversimplify brain chemistry, think of glutamate as the brain's gas pedal. It excites neurons and drives activity. GABA, gamma-aminobutyric acid, is the brain's brake pedal. It inhibits activity and calms the system down. T. gondii infection has been shown to alter the delicate balance of these neurotransmitters, often suppressing GABA.
Okay, so if you flood a brain with dopamine increasing reward-seeking behavior and simultaneously suppress GABA, removing the brakes.
You have a neurological recipe for severe impulsivity.
So if you have hundreds of these cysts in your brain pumping out excess dopamine and suppressing your neurological brakes, what does that actually do to a human being's real-world behavior?
Well, Jaroslav Fleger, the Czech biologist we mentioned earlier, spent decades running exhaustive psychological and behavioral profiles on individuals infected with T. gondii versus uninfected control groups. He found a consistent, measurable pattern. Infected individuals consistently showed subtly delayed reaction times and a significantly higher propensity for reckless, impulsive behavior.
And they tested this in the real world, right?
To test the real-world implications of this, independent epidemiological studies were conducted in Turkey and Mexico. They examined the blood of drivers involved in severe traffic accidents. The data corroborated Fleger's findings. People with T. gondii antibodies are significantly more likely to be involved in severe car crashes and industrial workplace accidents than uninfected people.
Their internal risk assessment software is being subtly corrupted by a cat parasite. They are making riskier overtakes, driving faster, or reacting just a fraction of a second too late.
Exactly. The parasite may be subtly trying to make the human host act more like the reckless rat, hoping to get it killed and eaten, even though humans are a dead-end host for the parasite.
Wow.
But the implications go much further than traffic accidents. There is a deeply disturbing, heavily researched link between T. gondii and severe mental illness, specifically schizophrenia.
Let's dive into that because the correlation is startling.
Decades ago, early pioneers like Otto Juravec and later Dr. E. Fuller Torrey began noticing a statistical connection. When you aggregate the global medical literature, people formerly diagnosed with schizophrenia are 2 to 3 times more likely to have antibodies indicating a T. gondii infection compared to the general population.
And Flager actually took this a step further by looking at the physical structure of schizophrenic brains using MRI scans, right?
Yes, a common physical anomaly found in many severe schizophrenia patients is a measurable reduction of gray matter in the cerebral cortex. Flager's team analyzed MRI scans of patients and found a chilling correlation. Almost every single patient in their study who exhibited that specific missing gray matter also tested positive for the parasite.
So the physical degradation of the brain tissue strongly correlated with the presence of the parasitic cysts.
Yeah.
We also need to talk about the suicide data in your research packet because it is incredibly sobering and points to a profound level of neurological manipulation.
Theodore Postolacha, a psychiatrist at the University of Maryland, began investigating chronic brain inflammation as a biological marker for severe depression and suicidal ideation. He teamed up with researchers in Europe to look at a massive prospective longitudinal trial.
Okay, what did they look at?
They analyzed the medical records of over 45,000 Danish women whose T. gondii antibody levels were recorded when they gave birth. Because this was a longitudinal study, they tracked these women over the next 15 years. The results were stark.
What did they find?
The women who had elevated T. gondii antibodies were 1.5 times more likely to attempt suicide during that 15-year period. And for the women who had the absolute highest levels of antibodies indicating a severe or active infection, the risk of a suicide attempt doubled.
Postolot didn't just stop with the Danish study, did he?
No, he sought to replicate the findings in different demographics. He conducted a similar study analyzing healthy residents in Munich, Germany. He found the exact same correlations. Higher T. gondii infection rates strongly correlated with clinical measures of impulsivity, thrill-seeking, and aggression directed either at oneself in the form of suicide or outwards at others.
Hearing all of this, it paints a very dark picture of human autonomy. But we have to ask the critical question looking forward. If this parasite is wreaking this much havoc, can studying it actually help us cure or treat mental illness?
Absolutely. That is the tremendous silver lining of neuroparasitology. For a century, psychiatry has struggled to understand the exact mechanisms of schizophrenia and severe depression.
Because the brain is basically a black box.
Right. But by mapping exactly how T. gondii physically manipulates dopamine production and how its proteins alter GABA and glutamate receptors, we are gaining unprecedented granular insights into the chemical mechanics of the human brain.
We're learning from the invader.
The parasite has spent millions of years perfecting a biochemical toolkit to control mammalian minds. If we can understand and reverse engineer the parasite's exact chemical toolset in neuroparasitology, could unlock entirely new classes of psychopharmacological treatments for mental health conditions that have baffled doctors for generations. We can learn how to fix the brain by studying how the parasite breaks it.
Okay, we have talked extensively about how a single parasite can alter one individual human's driving habits, their risk assessment, or their mental health. But let's zoom out.
Let's do it.
What happens when you multiply that subtle psychological manipulation by entire populations over thousands of years of human history? How does the invisible presence of pathogens shape human civilization itself?
This brings us to the fascinating intersection of evolutionary biology, sociology, and geopolitics. To understand this, we have to return to the behavioral immune system we discussed earlier, the subconscious feeling of disgust designed to keep us away from contagious germs.
Right, the evolutionary alarm bell that makes us avoid murky water, rotting food, and sick people.
Well, an evolutionary psychologist named Mark Schaller took that concept a massive step further. He hypothesized that this subconscious hardwired fear of disease makes us inherently prejudiced.
Prejudiced how?
If your brain is acting like a hypersensitive smoke detector for contagion, it doesn't just ring the alarm for obvious signs of sickness. It rings the alarm for anything that deviates from our evolutionary prototype of normal. Schaller argued that the brain's logic is crude but effective. If someone looks physically different from my healthy tribe members, they might be carrying a novel disease. Therefore, avoid them.
So simply because someone looks different, the brain misfires the disease avoidance alarm.
Precisely. Schaller conducted numerous psychological studies showing that when people are primed to feel threatened by disease, even subconsciously, they become measurably more prejudiced. They display higher levels of xenophobia, and they react with more hostility or avoidance toward the physically disabled, the obese, the elderly, and people of foreign descent. The brain is conflating physical difference with infectious threat.
There was an amazing, almost unbelievably simple experiment in your notes that proved how pliable and chemically driven this prejudice is.
Yes, it is a landmark study in behavioral psychology. Researchers took a group of participants and explicitly reminded them about the looming dangers of the upcoming flu season. As predicted, questionnaires showed this immediately spiked the participants' negative attitudes and xenophobia toward immigrants and outgroups.
So the disease threat elevated their prejudice.
But then the researchers introduced a variable. They gave half the participants a simple alcohol-based antibacterial hand wipe and had them clean their hands.
And what happened?
Just the physical act of disinfecting their hands, sending a sensory signal to the brain that the immediate biological threat was neutralized, completely erased the spike in prejudice. Their scores returned to baseline.
They literally washed away their xenophobia with a wet nap. It is incredible and deeply humbling how tied our complex moral and social judgments are to our primal sense of physical hygiene.
Which leads directly to the most controversial, sweeping, and fascinating theory in this entire field. The parasite stress model. This was developed by evolutionary biologists Corey Fincher and Randy Thornhill.
What did they look at?
They looked at massive amounts of global epidemiological data and asked a provocative question. Can the historical prevalence of infectious diseases in a specific geographic region accurately predict that region's modern culture, politics, and social norms?
And the data they aggregated was staggering.
They found a massive, undeniable statistical correlation. They looked at regions with historically high pathogen loads like tropical areas near the equator where the warm, humid climate allows parasites, bacteria, and mosquitoes to thrive year-round.
Okay.
They found that societies developing in these high-stress zones universally developed highly collectivist cultures. To survive the constant crushing threat of plague and infection, these societies became heavily reliant on strict traditions, absolute in-group loyalty, and extreme xenophobia.
That makes perfect evolutionary sense. I mean, if you live in an ancient village in a tropical zone and a stranger from the next valley comes to trade, they might be carrying a novel parasite that your tribe has no immunity to. That one stranger could wipe out your entire family.
Exactly. So cultural norms evolved to mitigate that risk. You don't marry outsiders. You don't trade freely with outsiders, and you strictly, sometimes violently, enforce local customs. Because breaking a cultural rule like how to properly prepare meat, how to dispose of waste, or how to wash before a religious prayer didn't just mean you were being rude. It literally meant you might catch a disease and die taking the tribe with you.
Thornhill and Fincher take this into the realm of religion and government too, right?
They argue that high parasite stress tightly correlates with the rise of autocratic governments, strict adherence to dogmatic religion, and highly conservative sexual mores. They point to ancient religious texts like the Mosaic Law in the Old Testament and view them through the lens of biology.
So religion as health policy.
Exactly. They argue these texts act as early, highly effective public health policies, disguised as divine mandates. Wash your hands before eating. Do not consume pork or bottom-feeding shellfish. Avoid contact with certain bodily fluids. If you do these things, you aren't just staying healthy, you are avoiding the wrath of God. Embedding public health in religious dogma ensures absolute compliance.
And conversely, what happens in regions with low parasite stress?
If you look at temperate zones with harsh freezing winters, which naturally kill off insect vectors and soil-borne pathogens every year, the parasite stress is much lower. In these regions, Fincher and Thornhill found strong correlations with the development of individualism, secularism, and democracy.
Because the biological penalty for interacting with a stranger wasn't instant death.
Right. People could afford to be mavericks.
Yeah.
They could explore trade freely and challenge traditional norms without risking the biological annihilation of their community.
So,
They even looked at something as seemingly arbitrary as right-handedness. In authoritarian high-parasite stress cultures, there is extreme societal pressure to conform to the norm because deviation is dangerous. Fincher and Thornhill found that these strict societies have significantly lower rates of left-handedness
let me guess they forced them to switch.
because natural left-handers are socially or physically forced from childhood to use their right hands to maintain absolute uniformity and conformity within the group.
Okay, I have to step in and push back fiercely on this model, because while the data correlation is interesting, isn't it incredibly reductionist? You are taking thousands of years of complex human history. The rise and fall of empires, the evolution of philosophy, economics, geography wars, the Enlightenment, and boiling it all down to where the bugs are.
It is a very fair, highly debated critique within the academic community.
I mean, to follow this logic, are we really suggesting that the 1960s civil rights movement or the sexual revolution only happened because we invented penicillin and suddenly felt biologically safe enough to be liberal?
Many sociologists argue exactly what you just said, that biological determinism ignores human agency entirely. And the researchers themselves, Thornhill and Fincher, acknowledge that human culture is multi-determined. Economics, geography, military history, and sheer historical chance play massive, undeniable roles.
So it's not the only factor.
We aren't stating that the parasite stress model is the only driver of history, nor are we validating one political system over another. But what researchers in this field forcefully argue is that deliberately ignoring the constant crushing evolutionary pressure of infectious disease leaves a massive, unexplainable hole in our understanding of sociology and geopolitics.
It's a missing piece of the puzzle.
The human brain evolved primarily to survive. Shaping our culture, our laws, and our religions to avoid disease was a primary, inescapable survival strategy for 99% of human history.
It is a lens that forces you to reevaluate every historical event.
Okay, so after spending an hour learning that invisible puppeteers are trying to dictate our politics, alter our societal norms, make us reckless drivers, and engineer our food webs, how do we actually protect ourselves? I want to make sure you, the listener, walk away with a practical framework for defending yourself.
The first step in defense is a shift in awareness. Building a framework for detecting environmental parasite risks doesn't mean looking for Hollywood zombies frothing at the mouth.
Right.
The parasitologist Robert Poulin points out that manipulations in nature are often incredibly deceptively subtle. You aren't looking for a wildly thrashing animal. You are looking for minor shifts in context.
Like what?
An animal being active at the slightly wrong time of day, or hanging out in an area with high visual contrast. Poulin cites Janice Moore's early experiments where she found that infected black pill bugs actively preferred to sit on white gravel rather than dark soil, deliberately making themselves perfectly visible to predatory birds. So the takeaway there is to be highly aware of organisms acting just slightly out of their ecological context. But what about actionable daily strategies for us humans living in modern society? Let's start with the elephant in the room, the family ad.
Right. It is vital that we do not induce panic and suggest you abandon your cat. While T. gondii absolutely relies on felines to sexually reproduce, epidemiological data shows that directly catching the parasite from a pet indoor cat is statistically much lower than other transmission vectors.
So we keep the cats.
If you have a cat, you simply manage the risk, ensure the litter box is scooped and cleaned every single day. The parasite eggs shed in the feces take 24 to 48 hours to sporulate and become infectious to humans. If you clean it daily, you break the cycle.
And wash your hands.
Also, ideally, the box should be cleaned by someone who isn't pregnant or immunocompromised. And keep your cat indoors so it cannot hunt infected mice and birds, which is how it catches the parasite in the first place.
The real danger zone for human infection isn't the litter box, though. It's actually the kitchen, isn't it?
Absolutely. The kitchen is the primary vector. You are statistically far more likely to contract T. gondii from eating undercooked meat. The parasite forms thick-walled microscopic cysts in the muscle tissue of livestock, particularly pigs, sheep, and cattle, that graze on soil contaminated by feral cat feces.
So ordering a rare steak.
If you eat a rare steak or saignant lamb, you are ingesting live cysts. Cooking meat to safe internal temperatures entirely destroys these cysts.
Good to know.
Furthermore, gardening without gloves or failing to rigorously scrub root vegetables like carrots and potatoes are massive risk factors. The soil in your garden is teeming with microscopic life, including parasite eggs. Wash your hands thoroughly with soap and water after touching soil because hand sanitizer does not kill parasite eggs.
What about risks when we travel globally?
A phenomenal example of mitigating global parasite risk is the campaign against the guinea worm. This is a horrifying parasite historically found in parts of Africa and Asia. You drink contaminated water, and a microscopic larva enters your body.
Oh.
Over a year, it grows into a worm up to a yard long inside your subcutaneous tissue. When it's ready to reproduce, it migrates to your lower leg and releases an acid that creates a massive, intensely burning blister. The pain is so severe and fiery that it biologically forces the human host to plunge their leg into a cool river or pond for relief.
Which is exactly the moment the blister bursts and the worm releases millions of larvae back into the drinking water supply.
Exactly. It is horrific, painful manipulation of human behavior to ensure waterborne transmission. But the defense against it is incredibly simple and elegant. We didn't need to invent a complex mRNA vaccine or a billion-dollar drug.
What'd it do?
The Carter Center spearheaded a campaign using simple, inexpensive water filtration. They distributed millions of low-tech pipe filters, essentially straws with a fine mesh cloth inside. Simply pouring drinking water through this cloth catches the tiny water fleas that carry the worm larvae. Thanks entirely to this behavioral intervention and low-tech filtration, the guinea worm has gone from infecting millions of people a year to hovering on the very brink of total global eradication.
That is one of the greatest public health victories in modern history. And finally, to close out our defense framework, we have to mention the intersection of sex and pathogens.
Yes. From a strictly biological standpoint, sexual reproduction is fundamentally risky behavior. It requires intense physical intimacy, the exchange of fluids, and the lowering of physical boundaries, which creates the absolute perfect bridge for parasites and viruses to jump from host to host.
Like rabies, right?
We see pathogens like the rabies virus
Mm-hmm.
actively inducing hypersexual or aggressive biting behaviors in animal hosts, specifically to spread through saliva. While we don't yet have definitive mapped proof of specific human sexually transmitted diseases chemically manipulating our psychological desire for multiple partners, the evolutionary logic of the behavioral immune system holds true.
Safe sex is an evolutionary shield.
Practicing safe sex isn't just a modern medical choice or a moral stance. It is an ancient, vital evolutionary shield against microscopic organisms that have evolved to exploit our most intimate biological imperatives.
We have covered an immense paradigm-shifting amount of ground today. We started by watching suicidal crickets hurl themselves into a glowing French swimming pool. We walked step by step through how microscopic worms act as massive ecosystem engineers, completely restructuring the food web and shuffling biomass up the chain to feed flamingos and pull endangered trout back from the brink of extinction.
We explored the relentless, multi-billion-year arms race between host defenses and parasitic cunning, diving into the Red Queen hypothesis and analyzing how our very sense of disgust serves as an invisible evolutionary alarm system.
We looked deeply into the mechanics of the cat parasite T. gondii, exploring how it performs drive-by chemical injections on our neurons, secretly hacking our dopamine and GABA levels, and drastically altering our risk assessment, contributing to traffic accidents, schizophrenia, and suicide. And finally, we zoomed all the way out to see how the invisible pressure of pathogens might have fundamentally shaped the laws, religions, xenophobia, and political structures of human civilization itself over millennia.
Processing all of this forces a complete humbling reevaluation of how we view human autonomy and our place in the natural world.
Which brings us to one final, deeply provocative thought to leave you with as you review your research. If your political beliefs, your religious fervor, your choice of a mate, your level of extroversion, your prejudice against strangers, and your daily risk-taking behaviors are subtly tuned by the microbial load in your environment, or by actual physical cysts quietly pumping out dopamine in your brain, where exactly do you begin and where does the parasite end?
It's a huge question.
Think about the legal implications. If a criminal on trial for reckless vehicular manslaughter is medically found to have a brain heavily infected with T. gondii, how should our justice system respond? Do we punish the human shell, or do we acknowledge the invisible passenger that was pressing the accelerator?
It is a profound, terrifying challenge to the very concept of free will and personal responsibility.
Next time you see a cricket in the forest, or a cat sleeping lazily in the sun, or even when you feel a sudden, inexplicable flash of disgust or a thrill of risky impulsivity, take a moment. Look at the world and your own impulses a little differently. You might not be the only one steering the ship. Thanks for joining us on this deep dive.